So we have 8 boxes for this part of our chapter on DNA. So what we're going to look at is the structure of DNA and how it's built. So the first part to talk about is that the monomer of DNA is called a nucleotide. And this nucleotide, I think the confusing part for students is that a nucleotide is actually made of 3 parts. It has the phosphate group, a 5-carbon sugar, and then a nitrogen base. So this one molecule right here is the monomer, even though it's made of 3 parts. And if you remember monomer and polymer, monomers join together to form long chains called polymers. And so when we look at this, here this 5-carbon sugar really looks like this. You can see later on in our video, we'll see why it's important to number these carbons. So 1, 2, 3, 4, and 5 carbons in this sugar. And so we call this deoxyribose. This is the name of the sugar in DNA. Now in RNA, it's also a 5-carbon sugar, but it's slightly different. Okay. Oh, there's RNA. So you can see the difference here. RNA has an extra oxygen, and so we call this deoxyribose. So basically without oxygen. All right. So we have the phosphate group, and if you remember phosphate groups from when we talked about phospholipids or ATP, they have that negative charge. And so we have our phosphate group as well as a nitrogen base. We have 3 different kinds of nitrogen bases. Oops, underneath my face you can't see, there's a C right there. So our monomer for DNA is made out of a nucleotide, or is a nucleotide, and a nucleotide is made out of a phosphate group, a 5-carbon sugar, and a nitrogen base. Ooh, and if we look at all of these structures, what we can see here is that our DNA is made out of carbon, hydrogen, oxygen, ooh, thanks plants, carbon dioxide and water, and then also nitrogen from the soil and phosphorus from the soil. So our, the elements that make up DNA, we have C, H, O, N, and P. Okay. Sorry. And so now let's take this monomer, this nucleotide, and you can see how it pairs together. So when we build DNA, DNA is a double helix, and it relies on nucleotides pairing together. We call this right here a base pair. A pairs with T. And so when we see this, the red little lines right here, those are hydrogen bonds. So the nitrogen bases are held together by hydrogen bonds. You can see the C and the G, though, while A and T have 2 hydrogen bonds, C and G have 3. Okay, so when we look at how our DNA forms, you can see how the middle rungs of that DNA ladder are held together by hydrogen bonds between base pairs. Now, what about right here, though, right? What about the bonds between the sugar and phosphate backbone? So when we talk about DNA, we call this area the sugar-phosphate backbone of the double helix, and we need to think about, like, the bonds between that. So right here, oh, look at, okay, I don't know if you noticed. So we're going to build some bonds in between the sugar and phosphate backbones. Now these bonds are called phosphodiester bonds. So we're going to go ahead and see a little bit more of how those are built. Oh, but before that, there's actually 2 kinds of nitrogen bases, and you can kind of tell by looking at them that the A and the T look different, and the C and the G look different. So when we classify these, we can see that purines have a double ring structure, and then pyrimidines have a single ring structure. Now, the way I remember this is that cytosine, and thymine both have Y's in them, and so does pyrimidine. And that's just the easy way that I remember it when I'm teaching. So when we look at DNA, and we were to line up our sugar and phosphate backbones, you can see right now that they're all pretty even. Now, if But you also notice, look at the center part. That is not quite as even. So if we were to look at what would happen if you had maybe a purine and a purine pair together, look at this carbon. Now it's too far off. Or if you were to have maybe two pyrimidines pair together, now this one's too far in. So when we look at like the Y of everything, purines and pyrimidines always pair together, because if not, you would have, it'd be too wide or too narrow, and the double helix would not work very well. Okay. So now, one reason DNA is awesome is because if you know one strand of DNA, you can figure out the other strand, all because DNA is complementary. Or complementary. I don't know how you say it. So if we know one strand of DNA, we can build the other, and this is important for DNA replication. So if we have one strand, if we have T, we know that A is going to pair with that, and then G pairs with C, A pairs with T, G pairs with C, C pairs with G. So we build our strands. Now if you notice, one side is upside down. Well, that's fascinating. Okay, so now let's go ahead and start the process of learning DNA replication. This video won't go into all the details, but at least prep us for class time. Okay, so now when we look at our 5-carbon sugar, it's important to number the carbons. And so when we look at this, we actually have specific places, like that's the 1, 2, 3, 4, and 5. So we call this the 3' and 5'. Those are going to be the ones that we are most interested in when talking about DNA and how it replicates. These are super important. We always need to pay attention to where's the 5' and where's the 3'. And so these are our ones that we are going to focus on. So when we look at our strands of DNA, you can see right here, this bottom part is the 3' end. And up here, this is where I'd find my 5' carbon, so I call this the 5' end. Now on the other side, I have my 3' end, and then my 5, ooh, I don't know what happened there. my 5' end right here. And so with this, we can see that our DNA runs in opposite directions. Complementary strands run in opposite directions, and we call this antiparallel. So the two strands of DNA are antiparallel and run in opposite directions. And we can see this by our 5' and our 3' ends. Okay, so when we look at two nucleotides, here's like the molecular structure of a nucleotide. Then I have another one. Ooh, and I want to build those phosphodiester bonds. Let's think about how this is going to happen. So I see a familiar OH and OH. Okay. And here we're going to remove a water and build a bond. Dehydration synthesis, just like we've learned for proteins and lipids and carbohydrates. So let me go ahead and just do that again for you. So you can see here we have our the top part. This is the 5' end of our nucleotide. This is the 3'. This is the 3' end of our nucleotide, and they join together by dehydration synthesis, and it forms a water. So you can see here is the phosphodiester bonding, the 5' end and the 3' end. Okay, so there's our phosphate groups. Here's our nitrogen bases, and here's our our sugars, and then here are our nitrogen bases. So you can see when we build our DNA, there's the 5' end, here's the 5' end, and here's the 3' end. So when we build our DNA, we always, this is critical, guys, we always are going to build and attach the 5' end to the 3' end. So we can only add bases to the 3' end. So when we build our DNA, here we're going to start with this nucleotide, and when we build our nucleic acids, we want to label first off, where is the 5', where is the 3'? So here's the 5' end, and the 3' end is down here at the bottom. And what's going to happen is I'm going to bring a nucleotide. Now you see this looks a little bit different than what we've been seeing. This kind of looks like ATP a little bit with the 3 phosphate groups. So it's kind of rad. So what happens is these nucleotides actually show up as nucleosides. As like, well, that's an S. As, oh my. Show up as nucleosides. And so they basically bring their energy with them. So they have 3 phosphate groups, and they're going to bring their energy with them, their own energy. And when they show up, what will happen is you're going to have the 2 phosphate groups are going to break off, and then ATP, like energy, will be released, and then that will be used to form this bond, the phosphodiester bond. So here you have, in the background, you see this blue circle or greenish, I don't know what color that is, circle called DNA polymerase 3. That's actually the enzyme that builds these phosphodiester bonds. So when you break off the 2 phosphates, there's energy that's going to be used to build that phosphodiester bond. And this is going to continue as we build that sugar phosphate backbone. All right. So DNA polymerase 3 is the enzyme that builds our strands. All right. So now let's go ahead though and look at the other side. So we see here that we have our 5' end and then our 3' end down at the bottom. So the other strand is antiparallel. With the other, oh, that is important. Good thing I put that arrow there. What we notice, this is important. Our DNA is built in the 5' to 3' direction. Always, always 5' to 3'. We built our DNA, in the 5' to 3' direction. We will never build it in this direction. It's impossible. And let me show you why. So now, when we look at that complementary strand, there's our 3' and here's our 5' end. So if I were to build it in the 3 to 5 direction, when that new nucleoside arrives, you can see that when the bond or the 2 phosphate groups break off, the energy is in the wrong place. There's no energy available to build that phosphodiester bond. You cannot attach it in this way. So instead, we're going to build it in the opposite direction. So we can go 5 to 3, 5' to 3'. And again, we're going to use DNA polymerase 3 to build these phosphodiester bonds. Now, when we do this, though, the one part that I have left off that we'll see in class is when we add our first nucleotide, like right here, we can't add it out of thin air. We need something called a primer. So just I want to put that in your heads that we actually do need a primer before we can begin to build DNA polymer. So, but I don't have that shown here. So anyway, so as we continue to build our strands in the 5' to 3' direction, we see a problem. Like what happens, here's DNA polymerase 3, and that's that enzyme that's going to add nucleotides in the growing strand. But what about this area, right? Where we can't build right here. And so what about this last phosphodiester bond? There's actually a different enzyme that's going to seal that for us. There's an enzyme called ligase. And ligase, oh, I like that. Want to see it again? So ligase is going to come and build that phosphodiester bond. So ligase is an enzyme that builds phosphodiester bonds. Sorry, I think, I don't know if I can move my face. Here we go. Is an enzyme that builds phosphodiester bonds between 2 nucleotides. And now we've built our DNA in the 5' to 3' direction. Oh, 5' to 3', 5' to 3', 5' to 3'. All right, good job. Oh, we built it in the 5' to 3' direction. All right, so this is just a cute little picture I thought was helpful maybe to kind of see that phosphodiester bonds forming there. Okay.